Hello everyone. The clinical enzymology, which is the last unit in our clinical biochemistry syllabus. We have discussed about so many diseases like diabetes mellitus, some liver diseases, some gastrointestinal disorders, some amino acid deficiency disorders, and some other diseases. And in this unit, the clinical enzymology, we are going to discuss about how the enzymes are used in the diagnosis of various diseases. So you have studied so many enzymes in the last 4 units, and in this unit you are going to study about the use of enzymes in diagnosis of some diseases. So what is the diagnostic significance of enzymes? So why the enzymes are diagnostically significant means it can act as a diagnostic markers of any underlying diseases. Enzymes can also act as reagents for various biochemical estimations and detections, and we have done some of the enzyme-oriented experiments in practicals also. So the enzymes as diagnostic markers, actually two enzymes are there based on its function. So one is functional plasma enzymes, and another one is non-functional plasma enzymes. So the functional plasma enzymes are derived from the blood plasma. So these plasma enzymes includes certain enzymes, proenzymes and their substrates which are present at all times in the circulation of the blood in normal healthy individuals. And these plasma enzymes perform several physiological functions in the blood of the healthy individuals. So for example, the lipoprotein lipase, pseudocholine esterase, and the proenzymes which are used for blood coagulation and the proenzymes which are used for blood clot dissolution are examples of these functional plasma enzymes. So the majority of these enzymes are synthesized in the liver and some of them are secreted by the liver. The next important group of enzymes are non-functional plasma enzymes. So these enzymes are cell-derived enzymes. So apart from the functional plasma enzymes, the plasma also contains some of the non-functional enzymes which contains numerous other enzymes that perform no known physiological function in the blood. So there is no notable function of these enzymes in the blood. So these apparently non-functional plasma enzymes arise from the routine destruction of erythrocytes, leukocytes, and other cells. So in general, any tissue damage or necrosis resulting from any injury or any particular disease is accompanied by increase in the level of several non-functional plasma enzymes in the blood. So just in this image, you see an example. A normal cell releases only a very few enzymes, whereas the cells, that is the B part, the cells which are affected by any disease or trauma condition, they release various intracellular enzymes due to the damage of the cells. What are all the possible mechanisms which are responsible for the increased enzyme levels in the serum? So the first thing, the increased release of enzymes may be due to the cell necrosis and also due to the permeability of the cell without any gross cellular damage. Sometimes the increased production of enzyme may also be noticed by means of overproduction of enzyme, so that one finally leads to overflow of the enzyme from the cell. So the increase in the tissue source of enzyme may occur in the case of malignancy. So apart from the increased release, sometimes the impaired disposition may also occur. So this condition will be there at obstructive jaundice, or sometimes it may be due to under the condition of renal failure. At the same time, the enzyme level will also be decreased due to two important reasons. One is genetic, that is from generation to generation, and another one is acquired, that may be due to any diseases. And sometimes it may also be due to the inhibition of any enzymes and also due to the lack of some cofactors. So the enzymes can be used in various diagnosis of diseases, and how this enzyme can be measured. That is the unit of enzyme. So the unit of enzyme activity can be measured by mostly accepted international unit, that is IU. And another one notation is also there, which is catal, that is the catalytic unit. So here in our country, mostly the IU is used. So one IU, that is the one international unit, is defined as the activity of the enzyme which transforms 1 micromole of substrate into a product per minute per liter of a sample under optimal condition and at a particular temperature. It is expressed as IU per liter. And another one unit for measuring the enzyme activity is catal, that is the catalytic unit. Here, one catal is defined as the number of mole of substrate transformed per second per liter of sample, and it is abbreviated as a cat or K. 60U equal to M cat and 1 N cat equal to 0.06U. These are all the units of enzyme activity. And what are all the clinical significance of enzyme estimation and how these enzyme estimation can be useful in assessing any disease. So single or serial assay of serum activity of a selected enzyme may help us in making the diagnosis, and it also helps in ascertaining the response to drugs in a diagnosis. Sometimes the response of a drugs can also be measured by measuring the enzyme levels, and the enzyme measurement may also help us to detect the disease at very earlier condition. It also helps in ascertaining the prognosis of a disease, and moreover mainly helps in ascertaining the time course of the disease, and we can know the stage of the disease by measuring the enzyme activity. The enzymes can be used as a diagnostic markers to detect various diseases. So several enzymes are helpful in diagnosing several kind of diseases. So these are some of the diseases which can be identified by measuring their enzyme activity. The first thing is the myocardial infarction, and the next one is liver disease. So the liver diseases can be measured by measuring so many enzyme activities like alkaline phosphatase and acid phosphatase. And the next one is muscle disease. And another one important disease is bone disease. And many of the cancers can also be diagnosed by measuring their various enzyme levels. And apart from all these things, many gastrointestinal tract diseases are diagnosed by simply measuring the enzyme activity. So with this, you just have an idea about enzymes which are used in diagnosis of various diseases, and we can discuss this in detail in upcoming lectures. Thank you.